Bismillahirrahmanirrahim, and welcome back to this next video. And in this video I am going to talk about an important family of the enzymes which are known as the GAPs. This is the video in connection to my last video in which I focused on another family of the enzymes which were known as the GEFs, the guanosine nucleotide exchange factor. And I've told you that the GEF, what they do is that when they act on their substrate protein, they are going to replace their GDP into a GTP. And I've told you that the substrate proteins for these GEFs, they are known as the GTPases. So these GTPases, that is a large family of the hydrolase enzyme that has the ability to hydrolyze this GTP attached to them into a GDP. What that means is that the GTPases, they can bind to the GDP, but they can also bind to the GTP. And the binding of the GDP would mean that the GTPases, they are in their inactive form, and when the GTP is attached to these GTPases, they are going to move in their active form. Now, as I've told you in my last video, that the GTPases has the ability to hydrolyze this GTP into the GDP to move from the active into the inactive form, but they cannot do it alone. They need the help of the family of the proteins which are known as the GAPs, which we are going to discuss today. So let us discuss about the GTPase activating enzyme. For short, we call them as the GAP. When you talk about these GAP, so this is the GTPase protein. We also call them as the G protein. So what you see over here is that when to these GTPases, a GTP is attached, this G protein can go for the slow hydrolysis to hydrolyze the GTP into the GDP, but this hydrolysis is not enough to move them from their active into the inactive form. And we need the activity of the G protein. We activate them. When we don't need their activity, we are going to move them into their inactive form. What the GAP are doing is that when the GAP, they get attached to these G protein or these GTPase protein, that is going to make the hydrolysis of the GTP very fast. Thereby, you can move the G protein from the active into the inactive form. So these are the G protein. They are also known as the GTP binding protein. And the important thing about the GAP is that they are only going to bind to the GTP bound form of the G protein. They do not bind to the GDP bound form of the G protein. What they actually do is they are going to bind to the GTP bound state of the G protein, thereby hydrolyzing their GTP into the GDP, thereby making them inactive. And this is actually the combined action of the GEFs and the GAPs, which are going to activate the protein when they are needed, and they are going to inactivate the protein when they are not needed. So this is what the GAPs they are doing. They are actually helping to increase the intrinsic GTPase activity of this G protein, thereby hydrolyzing the GTP very fast, thereby moving them from the active into the inactive form. Let me give you some examples of the GAPs. In my last video, I gave you the examples of the RAS GEF. So if there is a RAS GEF which can replace the GDP on the RAS protein into the GTP, so what the RAS GAP would do, that when it binds to the RAS GTP form, that is going to hydrolyze the GTP into the GDP, thereby moving the RAS protein from the active into the inactive form. So the RAS GAP is going to act on the RAS, and this RAS is a very well-known GTPase involved in cell growth and differentiation. Now, imagine the mutations in the RAS protein. The mutation in the RAS protein, if that is going to make it active for a very long period of the time, that can lead to oncogenesis, because this is very important in the cell growth. So the cell growth, when that is increased, that can lead to the oncogenesis. Another important family of the GAPs, and they are known as the Rho GAPs. And these Rho GTPases, they are very important family, like they are involved in the actin cytoskeleton remodeling, the cell migration, and the cell cycle progression. So these are two of the important examples of the GAPs. The RAS GAP, they are going to act on the RAS protein. The Rho GAPs, they are going to act on the Rho family GTPases. Now, why this is important? Because when you talk about these G proteins, they are involved in a variety of the cellular signaling processes like the signal transduction, the cell growth, and differentiation. What these GAP do, that they play an essential role in controlling the duration. Now, this is very important, that for how much time you need the activity of a particular GTPases. If you are increasing that particular time, that is going to have negative consequences on the cell and of course on the body. And the intensity of the signals by acting as the negative regulator. So the GAP is actually a negative regulator of these GTPases, and this negative regulation, that is very important to control the duration and the intensity of the GTPases. Now imagine the dysfunction in the function of the GAP, that can lead to various diseases like the cancer, cardiovascular disorders, and the neurodegenerative diseases. So if I focus on this one, the rest is involved in the normal cell division. The normal rest is involved in the normal cell division, but if there is mutation in the GAP, and that cannot control the duration and the intensity of the rise signal, that may lead to abnormal cell division, that can lead to the cancer.